Hello all, my name is Sarah Mills. I'm a pharmacy resident at Moses Cone, and together with my co-resident Mimi Pham and a previous resident Kinney Kong, we will be presenting you with your first lecture, An Intro to Pharmacology. We'll cover topics of pharmacokinetics, dynamics, genomics, economics, and the FDA approval process. This is a very high-level 1,000-foot view, so this is a great place to start when you're learning pharmacology, and you'll have additional materials in this unit on your Canvas site. Our objectives for today are to define pharmacokinetics and pharmacodynamics, to describe how pharmacogenomics affects drug response. We'll learn how pharmacoeconomic principles influence care. We'll gain a greater understanding of the FDA approval process. And by the end of this lecture, you'll be able to apply basic pharmacology principles to patient cases. The first three topics we will be discussing are pharmacokinetics, pharmacodynamics, and finally pharmacogenomics. These three scientific principles are all working simultaneously to determine drug response for each patient. Very simply, kinetics is what the body does to the drug, dynamics is what the drug does to the body, and genomics is how DNA and gene expression influence both of these domains.
So first is pharmacokinetics. So first for absorption, there are four key concepts I want. Next, we'll talk about distribution. So four main factors that affect this. Next is metabolism, and there are four. And finally, the last concept of pharmacokinetics I will discuss is a limit. So next, we'll move on to the topic of pharmacodynamics. Another concept of pharmacodynamics another concept here is affinity. So how tightly and to what extent. Two related concepts, potency and efficacy. Two more terms I want y'all to be familiar with are tolerance and dependency.
One thing I was asked to cover during this lecture is race-based effects on pharmacokinetics, dynamics, and genetics. It's important to understand that this topic, in an overview lecture, there's not enough time to possibly scratch the surface of all the ways that race impacts medical care. So I'm going to give you a brief overview with some considerations when encountering race-based medicine, because it is something that will come up during your practice. So I think it's important to cover a few examples and a few important points. Just know that this is meant to be an overview and not meant to be comprehensive in a way that this topic really deserves. If you have specific questions, feel free to forward them to me prior to class so I have the relevant data ready to share. And I will include some references that you can look up for your own reading on each slide. The first thing to consider is race versus ancestry versus genetics. We know that race is not predictive of ancestry, and ancestry is really the proxy that we would consider when looking at genetics, and genetics is what would really affect drug response. Racial essentialism believes that races are biologically different groups determined by genes. What contradicts this is that the genome between socially constructed racial groups is 99.5 to 99.9% identical, and there are no identifiable racial genomic clusters. Race continues to be used as a biological reality in health disparities research, medical guidelines, and standards of care. This reinforces the notion that racial and ethnic minorities are inferior while ignoring the health problems of historically privileged groups. The real truth is that racial differences found in large data sets most likely often respect health disparities as a result of racism and not true genetic differences. One example of historical race-based medicine is creatinine clearance calculation. Persons with assumed African American heritage were thought to have more muscle mass, so an arbitrary correction was applied to calculate their kidney function. This led to an underdiagnosis of kidney dysfunction in this population. The most currently accepted and recommended formulas, for example CKD-EPI, shown here on this slide, do not include race as a predictor of kidney function, leading to more accurate kidney dysfunction diagnoses. Looking at articles from around 1995, it was assumed that race caused differences in bioavailability, protein binding, volume of distribution, hepatic metabolism, and renal tubular secretion of medications. When this data was reconsidered, it was evident that other patient-specific factors like diet and supplement use could have more heavily impacted this data. When considering prescribing medication, diet and supplement use are valid things to consider, but we shouldn't assume patient-specific factors based on race. When looking at ancestry, there are real screening considerations, such as screening for rare diseases in an Ashkenazi Jewish population, or considering that the HLA-B star 5701 in persons of East Asian descent is rarely present, but it's a highly prevalent genetic variant in persons of Southwest Asian descent. This is important to screen for before administering abacavir because it can cause life-threatening hypersensitivity reactions. Genetic differences are really what is valid to consider when evaluating the effects of ancestry on the way we practice medicine. This is a great transition into pharmacogenetics and genomics, which is how DNA and gene expression influence the kinetics and dynamics of a drug. Practically, these words are used interchangeably, the pharmacogenetics and genomics, so you don't have to really worry, but in case you want to know, pharmacogenomics refers to the role of various components of the genome in response to a drug, so the DNA and everything that affects its expression, and pharmacogenetics is a subcategory of that just refers to the role of genetic variation in response to a drug, so just alterations in the actual DNA sequence.
The majority of CYP genes encoding for enzymes are polymorphic, leading to genetic variation. These polymorphisms can contribute to diminished or absent metabolism or excessive metabolism of a compound. These are described as ultra-rapid, extensive or normal, or poor metabolizers. For substrates that are metabolized into inactive forms, ultra-rapid metabolism will cause less active drug because it's being quickly deactivated, which can reduce the efficacy of a drug. Extensive metabolism results in normal amounts of active drug, and poor metabolism leads to less deactivation and thus more active drug, which can cause increased sensitivity and possibly toxicity. Now, this is opposite for drugs with active metabolites. Ultra-rapid metabolizers will then lead to more active drug. Extensive metabolizers will again produce the normal or expected amount of drug, and poor metabolizers will lead to less active drug. This is important to consider especially for prodrugs that must be metabolized to an active form in order to produce its desired pharmacologic effect. The most clinically important polymorphic variations involve enzymes CYP2C9, 2C19, 2D6, and 3A4. And just as a quick review, I have listed the breakdown of the nomenclature here. The first symbol indicating the family, second, the subfamily, third, the isoenzyme, and the fourth and possibly most important piece when referring to pharmacogenetics is the allele, or where we will see polymorphisms. Starting off with CYP2C9, I've listed some of the most common drugs affected here. Examples of common substrates include phenytoin and warfarin. Inhibitors include SSRIs, and common inducers carbamazepine and phenytoin. For this presentation, it's most important to remember the substrates. As I mentioned, warfarin is one of the most commonly affected substrates. It's a racemic mixture in which the S enantiomer, which is the active form, is metabolized by CYP2C9. Alleles 2 and 3 are polymorphisms that indicate poor metabolizers. So again, with poor metabolism of the active form, these patients will have increased activity and may require lower doses to achieve desired INR. This also means that if the dose is not lowered, patients can experience undesired side effects or increased risk of bleeding. The FDA actually now recommends testing for patients on long-term warfarin, though this is not always done in practice. CYP2C19 is another enzyme with significant polymorphisms. Clopidogrel and phenytoin, again, are common substrates. Inhibitors include fluconazole and PPIs, and common inducers include rifampin and carbamazepine. Clopidogrel is one of the weird ones, since it's a prodrug that is metabolized by CYP2C19 to an active form. Phenotypes 2 and 3 are poor metabolizers, which leads to less metabolism into the active form and lower efficacy, but we can with this see increased cardiac side effects as well. Phenotype 17, on the other hand, indicates an ultra-rapid metabolizer, so increased metabolism to the active form and thus increased activity. Some polymorphisms are more common in specific patient populations. Patients of Asian descent, for example, are most commonly found to be poor metabolizers and often need increased doses or even alternative therapy when using clopidogrel. CYP2D6 is most commonly associated with antidepressants and opioids, but I've listed common substrates and inhibitors on this slide as well. Though it's not always part of major metabolic pathways, it is a minor enzyme for many, many drugs. It's found to have some of the highest degree of genetic variability, with over 75 identified variants. The majority of these code for poor metabolizers, with at least 22 loss of function alleles, where the enzyme has just that, complete loss of function, and is unable to perform any metabolism at all. Though the most common polymorphisms are found within the CYP enzyme family, we can also find differences in phase 2 metabolism. An alteration in uridine glucuronol transferase, or UGT, can code for reduced expression and function, which can be seen in the metabolism of irinotecan, for example. N-acetyltransferase polymorphism include NAT2, which code for slow acetylators. Isoniazid and hydralazine may be affected by these alterations and can result in increased side effects due to reduced metabolism to inactive or non-toxic forms. I want to go through a quick patient case to demonstrate how pharmacogenomics can be used in practice. A 38-week-old male was delivered with no issues. After 1 week of life, the baby was still having difficulty breastfeeding, along with presenting with increased lethargy. On day 11, he was seen at his PCP for his well baby visit, and the next day, the family reported he had gray skin and decreased appetite. On day 13, his health worsened, and he was found non-responsive secondary to respiratory depression. An investigation after his death showed that the mother was prescribed codeine 30 milligrams plus acetaminophen 500 milligrams for post-episiotomy pain. She was initially taking 2 tabs every 12 hours, but by day 2 she was experiencing some somnolence and constipation, so it was recommended to decrease her dose to only 1 tablet. Postmortem analysis of the infant showed no anatomical abnormalities, but did show a morphine blood concentration of 70 versus an expected level of less than 2. Codeine normally undergoes multiple metabolic pathways. Normally, glucuronidation converts 50 to 70% of the compound to less active form, codeine 6-glucuronide. 10 to 15% is converted by CYP3A4 to norcodeine, and finally the remaining 5 to 15% is converted to morphine by CYP2D6 before undergoing glucuronidation for eventual elimination. With genotype analysis, investigators stated the mother was found to be a CYP2D6 ultra-rapid metabolizer, meaning a greater amount of codeine was converted to morphine. In addition to this, infants do not reach adult levels of glucuronidation until about 3 years of age, so a significant amount of morphine was unable to be further metabolized into its inactive form for elimination, which resulted in abnormally high levels of morphine, ultimately contributed to the baby's death. Pharmacogenetic testing is meant to guide treatment strategies, patient evaluations, and decisions based on its ability to predict response to treatment and avoid situations like the patient case we just discussed. When testing is targeted to evaluate an individual's response to a specific drug, typically one gene is analyzed. However, laboratories have developed multi-gene panels that include more than 2 genes in order to proactively evaluate an individual's response to many drugs. The most common genetic testing is used for drugs that are used to treat anxiety and depression, such as SSRIs, pain, and cardiovascular disease, like I mentioned, warfarin earlier. Though pharmacogenetics shows promise and data supporting its use is growing more and more each year, there are many challenges to its use in clinical practice. There are limited studies with varying designs focusing on particular patient populations and limiting its overall use on a universal standpoint, as well as a lack of cost-effective analysis. There are significant regulatory and ethical concerns, and because of this, there are no true guidelines for testing or implementation, as well as a lack of education for both clinicians and patients. And with all of these things put together, there are often risks of delays in therapy because of the uncertainty that surrounds pharmacogenomics.
So that will cover our conversation related to pharmacodynamics. Next, we'll go into pharmacoeconomics briefly, and this is broadly defined as the analysis of costs and consequences of any healthcare-related treatment or service. And you see 5 different types of analysis here, and we will go into this. Really, the point of explaining what pharmacoeconomics to you in this setting is to address the fact that costs do play a role in healthcare. It's very expensive to be in the hospital. It's very expensive for a lot of different medications, and we just want to bring your attention to how you may be able to help the costs of your patients moving forward.
So within pharmacoeconomics, there are a multitude of factors that affect how we analyze the cost, the efficacy, and how we conduct these type of studies. So first, we always want to look at the clinical efficacy and its effect on the patient, as well as the safety aspects of the treatment. We also want to look at adverse drug reactions. We want to look at hospital admissions and whether a treatment can help or reduce hospital admissions, as there are costs tied to those. We want to assess the patient's quality of life. We're going to look at different economic outcomes, such as costs, including direct, indirect, and intangible costs. We want to look at the total cost of the treatment itself directly, as well as any ancillary treatment and other associated costs with the main treatment, and a multitude of other factors as well that can be included in various pharmacoeconomic studies. So one important thing with pharmacoeconomics to consider that is a little different than some of the scientific literature that you may have seen in the past in your experience as nurses is the point of view. So in our example, there are 3 main point of views that pharmacoeconomic studies can come from. The first being the payer. These are your insurance companies and your government payers, such as Medicare and Medicaid. They're going to look at the economic value of a treatment or a test a lot differently than a provider, someone who's taking care of the patient and providing the frontline care. And they still even may look at it differently than a patient, the patient who ultimately has to bear the brunt of the cost. And each patient may value something differently as far as what a treatment brings to them versus the adverse effects that it also brings to them. And then in addition to those points of views, the costs that we look at in pharmacoeconomic studies are also very important. Obviously, there's always direct costs. What's the exact cost of the drug? What's the exact cost of the test that the patient or the insurance is going to have to pay for? But then there are a lot of indirect costs, as well as intangible costs, costs that we just can't truly measure. And we'll discuss some of these moving forward. So with pharmacoeconomics, there are 5 different types of studies and analyses that we can do. The first is a cost of illness analysis. This is a very general analysis looking at the total cost of any disease within a population. So for example, if we wanted to look at heart failure patients, maybe we would tie up all the intangible costs, the indirect costs, and the direct costs of all of our heart failure patients in the United States, and that would be the cost of illness. Second, we have cost minimization analysis. This is an analysis that you can do when you assume that 2 different treatments are equally as efficacious as each other. In this way, what we do is we assign costs to the treatments and essentially assess which one is cheaper, knowing that they are going to produce the same effect. For the next 3 types of analysis, the cost effectiveness, the cost benefit, as well as the cost utility, it is best to think about how the analysis is analyzing the cost side of things as well as the benefit side of things. So here's what I mean by that. For cost effectiveness, you're essentially looking at treatments that you don't necessarily think are effective. For the costs, you can just use dollars as your unit of measure, and how much does it truly cost for that treatment and for that test. But for the benefits, you may use a different parameter. You may use a unit of measure that is directly related to the disease state. For example, hypertension, we may choose to use the decrease in systolic blood pressure. And through that, for each treatment, we can decide how much does it cost per 1 unit decrease of the systolic blood pressure, and that would be a cost effectiveness analysis. For cost benefit, what we're looking at is that the cost side is in dollars and we want to assess the benefit in dollars. This is a little bit more difficult as benefits are often perceived, and trying to say that I feel so and so better, or I feel my blood pressure has decreased by so and so amount, doesn't necessarily translate exactly to a dollar amount. However, we do our best within these studies to figure out appropriate translation. And finally, cost utility analysis are looking at cost again in dollars, but the benefit this time is measured in utility, or quality adjusted life years. And this quality adjusted life year is often referred to as QALY, Q-A-L-Y, is a measure of the quality of life that the patient is having. Again, this is a difficult measure to try to assess since it is perceived and can vary very much from patient to patient. But it does present a different outlook on how we are measuring pharmacoeconomics as not just a dollar amount, but truly how is the patient feeling and how is the patient living day to day.
So for parts of Medicare, there are four main parts. Part A is known as the hospital coverage. Part B is the medical coverage. Part C is Medicare Advantage. So this is usually a supplemental on top of Part A. And then Part D, which is easy to remember for D, is for drugs. So this is your prescription coverage. There are further details for each part of the Medicare coverage on here, but the most common ones that we will see as far as pharmacy would be Part D, of course.
So that briefly covers the main types of studies and analyses that can be done in pharmacoeconomics. Again, this is a very surface level view. We can discuss deeper in class if you have more questions. But it gives you a general idea of how in healthcare we try to evaluate the costs that are associated with the various treatments we have. And even though it's a touchy subject, it's something that it is important to consider to make sure that we bring the best value to our patients. So next we'll briefly talk about Medicare coverage. Medicare is the main federal government payer for adults over the age of 65. Now that age can change in the future. And it does cover some other special populations as well. And the coverage can change from year to year. Now Medicare is divided into different parts. There's Part A, Part B, Part C, and Part D. For the purposes of this class, it is important to know that for drug coverage, that is Part D, D for drug. What's interesting about the Part D coverage, though, and this is something that you'll see a lot with your patients when you start prescribing medications, is that there's this thing called the donut hole. So as with any insurances, different plans may have deductibles and co-pays and other things that are coming out of the pocket from the patient, whereas the insurance itself, in this case Medicare, can cover and pick up the rest. However, at a certain dollar amount that can change from year to year, there is this thing called the donut hole. Essentially, when you reach that certain dollar amount that you've spent on the year, the insurance is no longer covering you. So you're paying everything out of pocket, and essentially your drug expense goes through the roof. And then that donut hole exists for a certain amount of dollars, and then if you then reach a second threshold, you can actually leave the donut hole and qualify for something called catastrophic coverage. Some patients may never leave the donut hole. They may reach the donut hole and reach that preset amount around September, and then they don't get out of it in December. So September through December, everything's out of pocket before it resets in January. Of note, the donut hole has been shrinking over the previous years, and the plan is for it to disappear in the coming year. So it's not something you may necessarily need to worry about when you actually get out of practice as nurse practitioners.
A common concept that you hear with patients with Medicare Part D is that they reach a donut hole. So during this donut hole, patients are actually responsible for 25% of their prescription costs, and they usually reach this donut hole once they reach a certain limit of their prescription costs, including what they pay for and what their insurance carrier pays for. And this certain limit isn't a fixed amount. It is determined by the carrier, and it does vary annually. So usually whenever patients reach this donut hole, they'll notice that their medication costs are much higher, but it's similar to kind of a deductible period, but it's not a deductible period. And then after they're out of the donut hole, their medications will return to the copay costs that were outlined in their original terms for their insurance. Okay, next we'll go through how we bring a medication to the market via the FDA with a drug approval process. Everybody is familiar with the FDA, or the Food and Drug Administration. They're responsible for bringing new medications to the market, but it's actually a particular branch of the FDA that is responsible for this, which is the Center for Drug Evaluation and Research, or the CDER. Their mission is to ensure that drugs that come to the market are safe and effective, and their primary role is to evaluate these new drugs. They regulate over-the-counter medications on top of prescriptions, and that includes brand and generic medications. So the overall drug approval process begins with a preclinical phase, which takes usually about a year to 2 years. This is where the drug sponsor is discovering and screening the medications. So during this time is drug development. They test the safety of it on animals or even effectiveness, and then they also submit a IND application to the FDA. And then moving on to the clinical process, which is longer, about 5 to 7 years. This is when the clinical trials are carried out, which most people are aware of. And then there's the NDA review occurring after the clinical phase. So once the drug manufacturer applies a NDA, this is reviewed, and this includes all the information for safety and efficacy that the drug sponsors gathered during their preclinical and clinical phases. So then after that, if their NDA is approved, it goes on to drug labeling, and then also a facility inspection of the site of where the medications are manufactured. And then once it gets finally approved through the FDA, afterwards there's a post-marketing phase, also known as phase 4. So when the medication actually goes onto the market, patients are prescribed it and they want to take it. This is during a time where the FDA are monitoring reactions or any kind of events that are related to the medication that are being voluntarily reported to the FDA. So if a patient takes a medication and it's a brand new medication to the market, the FDA usually the patient would have to tell the FDA if there are any side effects, or the pharmacy or the hospital or any healthcare system would be the ones to report these side effects that were not caught during clinical phases to the FDA. So looking further in detail of the clinical phase of the drug approval process, again, this is where we have the clinical trials that most people are familiar with. There are 3 main phases. So phase 1 is the smaller initial phase where we start with healthy patients who do not have the disease. And the main purpose of this phase is to test the safety of the medication. Then phase 2 is a little larger, and this is a group of patients who actually have the disease that the medication is designed to treat, and this is to test effectiveness of the medication. Then finally, for phase 3, this is a much larger, sometimes a landmark trial with diseased patients, and this is to test the safety and efficacy of the medication. So once the medication is FDA approved and is on the market, patients are able to take this now and it's being prescribed. The post-marketing phase, again, is phase 4 of clinical trials, and it goes on for years after the medication is already on the market. So the purpose of this phase is that since it's not possible for the clinical trials to catch every drug effect or side effects that the medication may incur, so this post-marketing phase is important for those side effects to be reported afterwards. And usually the reporting can occur, anybody can do the reporting. Again, this is a voluntary process. You can report to FDA's MedWatch program. This is the patients can do it, general population, or even the providers will do it. And then usually what the FDA would do from this, they would determine if the side effect is, or how detrimental the side effects are, and if they should remove the medication from the market or not. This is a timeline example of the drug approval process for a very common medication, amlodipine or Norvasc for blood pressure. And you can see that this medication has actually been on the market for a long time, but the preclinical the preclinical phase of this medication, or when this medication was actually discovered, was back in the 1960s. And you can see that it took some time for them to complete the clinical trials for this medication. And then it was finally approved by the FDA in 1995 or 1996, and then ever since then, and still going on now, there are constant post-marketing monitoring with this medication, just again via MedWatch. So this overall process for when the medication was discovered until it was FDA approved and put onto the market took 15 years, and this is a very traditional or most common way that medications are approved now. We also wanted to provide an example of a non-traditional drug approval process. So given our current healthcare state right now, we wanted to discuss Pfizer. So the Pfizer COVID-19 vaccine, or now brand name Comirnaty, was actually approved by the FDA last year in August. But prior to that, starting in 2003, there were already research projects or discovery processes for vaccines around SARS and MERS. And then from there, they developed further vaccinations around SARS-CoV-2 virus once that genetic code was available. And then due to our pandemic, the FDA actually sanctioned this to be emergency use or available for emergency use. And since they were able to give this as an emergency use, the monitoring for safety and efficacy of this new COVID vaccine started then in 2019. So the company actually bypassed having to apply for an NDA, and with the FDA being able to already monitor the safety and efficacy while this medication was being provided as an emergency use, they were able to get it approved last year. And right now, there's still ongoing post-marketing monitoring for this medication or for this vaccine.